Hello everyone, welcome to 10-Minute Physiology. In today's video, we're going to look and see how we have color vision. So with that, let's give it a go. So the first thing that we have to understand is what an absorption spectrum is. So an absorption spectrum is basically a graph that describes to you the probability of a particular wavelength being absorbed by a pigment. So what we're going to do in this video is we're going to look at the absorption spectrum for each of our photoreceptor cells, so for our rods and for our 3 cones. And what we see here on this graph is 2 axes. On the y-axis we have the relative absorbance, and on the x-axis we have the wavelength of light in nanometers, going from the violet region all the way to the red region. So let's take a look at our first absorbance spectrum, and it looks something like this. So this first absorption spectrum belongs to a photoreceptor cell. And what we see here, first of all, is that this particular photoreceptor cell can absorb light over a wide range of wavelengths. In fact, it can absorb light all the way from the violet region to the green-yellow region. However, what we see here is that there is a peak absorbance, and this peak absorbance is equal to 420 nanometers. And 420 nanometers is within the violet to blue region of the visible spectrum. So what is the peak absorbance? So the peak absorbance is the wavelength of light in which the pigment has the greatest probability to absorb photons. And in this case, this photoreceptor's pigment has a peak absorbance equal to 420 nanometers, which is within the violet to blue region of the visible spectrum. And it's because of this that it has a peak absorbance within this region of the visible spectrum that it's called the S cones or blue cones. So this particular absorbance spectrum belongs to the S cones. So the next absorbance spectrum we'll look at is this one. And what we see here is that it has a peak absorbance equal to 475 nanometers, which is within the blue to green region. Now this particular absorbance spectrum doesn't belong to a photoreceptor cell in this case. Instead, it belongs to a protein called melanoopsin. So melanoopsin is a protein that we're not really going to talk about in this video, but we'll talk about it in the next one. So just keep it in the back of your head. So the third absorbance spectrum is this one, and what we see here is that it has a peak absorbance of 500 nanometers, which is within the green region of the visible light spectrum. This particular absorbance spectrum belongs to our rod cells. So the fourth absorbance spectrum that we're going to look at is this one. And what we see here is that this particular absorbance spectrum has a peak or absorbance of around 530 nanometers. This is going to be within the green to yellow region of the visible spectrum, as we see right here. And it's because of this we call them the M cones or green cones. So this particular absorbance spectrum belongs to the M cones. So the last absorbance spectrum is this one. It has a peak absorbance equal to around 560 nanometers, which is within the yellow to red region, and this is going to belong to the L cones. So this right here are the absorbance spectra for all of our rods and cone cells in our retina. And the main point that I want you to remember here is that even though each of these particular photoreceptor cells has a peak absorbance, all of them can absorb light from a wide range of wavelengths. So just as a reminder, let's summarize the peak absorbances for each of our cone cells. So for our cone cells, what we see here for our S cones is that they had a peak absorbance or wavelength of around 420 nanometers. The M cones had a peak absorbance equal to 530 nanometers, and the L cones had a peak absorbance equal to around 560 nanometers. So now that we have that under our belt, let's explain how we have color vision. Well, the first thing to note is that humans have 3 different types of cones. We have M cones, S cones, and L cones. And as we saw previously, each of these cone cells has a different absorbance spectrum. Now, it's important to realize that a single cone cell does not encode the wavelength of a light stimulus. If a cone cell responds to a light stimulus, the cone will generate the same response regardless of the wavelength. And the reason why is because the pigments in each of our cone cells can respond or absorb light from a wide range of wavelengths, as we saw. However, the pigment is going to be most likely to absorb the photons when their wavelength is at the peak absorbance. So remember that our graphs for each of our individual cone cells, they overlap a wide range of wavelengths. So these cone cells can absorb light from a wide range of wavelengths, which means that they can absorb light from many different colors. And it's because of this that a single cone cell is not able to determine what wavelength we're actually seeing. However, light that hits the cone on the edge of its absorbance range can still generate a large response if the intensity of light is high. So in other words, even if there is a very low probability of a cone cell absorbing a particular wavelength of light, it can still generate a very large signal if the intensity of that light is very high. Therefore, it's because of all of these things, if we have only one type of cone in our eyes, our vision would be monochromatic. However, as you know, our vision is not monochromatic. We're able to see many different colors. So how can we see in multicolor? Well, the reason why is because we don't have a single type of cone. We have three cones working in our eyes. We have our L cones, we have our M cones, we also have our S cones as well. So all of these cones are working together in order to receive light, and they're all connected in our retina and sending signals to our brain. So in order to understand how we can decode a particular wavelength, let's do a little experiment. So we have all three of these cones wired to a brain. And let's just say we release monochromatic light being red. So this is red light being released onto these three cone cells. So what would happen? Well, this light that is released from the environment onto these three cone cells would cause these cells to react. So let's just say after this cone absorbs the red light, it produces three stars, which correlates to a specific signal. When the green cone here absorbs the red light, it also produces a signal. However, in this case, it's symbolized by two stars. And the blue cone here, let's just say, only produces one star, which represents a slightly different signal. So in other words, a light of a single wavelength stimulates each cone to a different degree. The nervous system then compares the relative stimulation of the three cone types to decode the wavelength. So in other words, the brain is comparing what the signal looks like from each different cone, and depending upon how these signals compare, it then can determine what the wavelength of light that we are seeing. So we need multiple different types of cones in order to decode the wavelength of light that we are seeing. So now we're going to finish off this video with scotopic versus photopic conditions. So here we have our absorbance spectrum graph again. However, this time, instead of looking at individual photoreceptors, we're looking at the eye as a whole. So let's just say we have an eye that is dark adapted, or in other words, it's been exposed to darkness for quite a long time. What would the absorbance spectrum for the retina look like? Well, it would look something like this. And what we see here for this retina is that the peak absorbance is going to be at 500 nanometers. And if you remember, which particular photoreceptor had a peak absorbance of 500 nanometers? Well, the answer was the rod cells. So what does this absorbance spectra tell us? Well, it basically tells us that under conditions of dark adaptation, or scotopic conditions, sensitivity to light depends mainly on the rods. And if you watched my previous video, that should make sense. Because remember that when our eyes start to become dark adapted, the threshold of activation for the rods drops really low, which allows them to work well in the dark. And that's why we rely upon our rods more than our cones when we're in the dark. And this is what this graph is showing us here. So what if we were to go into the light? Well, if we were to go into the light, the absorbance spectrum would look something like this. And what we see here is that the peak absorbance is equal to 560 nanometers. So what is this telling us? Well, under conditions of light adaptation, or photopic conditions, sensitivity is mainly going to depend upon the cones. And this should also make sense from my previous video. And this is because the cones basically have a higher threshold than the rods do, so therefore they work better under conditions of light. So what do we get in general from this graph? Under scotopic, or dark adapted conditions, the eye is going to be most sensitive to light at 500 nanometers. Under photopic conditions, which is under light conditions, the eye is most sensitive to light at 560 nanometers. So that's it for this video. I hope it helped you understand how we have color vision, and I hope to see you in the next video. Thank you for watching, and I hope to see you next time.